Okay, okay.
And we are live.
Okay. So we will need another minute before we begin. Pramod sir is just joining us back.
Yeah, I'm here.
Right. So we will begin, sir.
Yeah.
Yeah.
Yes.
Hi everyone, my name is Varidhi. I'm from Careers 360 and I welcome you all to this session on careers in biology. With me is my colleague Gauri Rane and we will be taking you through the session for today. So we know that biology is held as a favorite subject by a large number of students and they want to pursue it in the long run. What often becomes a hindrance is a lack of awareness about what kind of courses and what kind of career prospects they can look at for themselves. So to answer all these questions for you, we have with us a very distinguished speaker for today, Dr. Pramod Yadav. Hello, sir. Thank you so much for joining us. A little more about Dr. Pramod before I request him to take over. So, PhD from Banaras Hindu University and former postdoctoral researcher at Institute of Molecular Biology, Zurich. Dr. Pramod's expertise lies in molecular biology. He was awarded Senior Research Fellowship by the University Grants Commission, that is the UGC, in 1976, dedicated to working in education in the life sciences. Currently, he is visiting professor at the Department of Biological Sciences, ISER, Behrampur, while he has been formerly Dean of the School of Life Sciences, Jawaharlal Nehru University, New Delhi. I would once again like to thank you for joining us today, sir. It's a pleasure to have you. And without much ado, I would request you to please take over and address our students. Post that, we will open the floor for questions and answers.
Okay. So I must be assured that my voice is reaching the students.
It is fine.
We
can hear you.
it
Can
should
hear
be
you.
okay. Yeah, it's wonderful to have this session addressing likely options in biological sciences. When you start up in a school or let us say intermediate level education of for biology, you are commonly looked at as someone who is opting for future pursuits in medicine or maybe an academic pursuit in botany, zoology, biochemistry. But that is not the full illustration of what biology today offers. Biology today is an all-encompassing discipline and requires skills from all fields of sciences: physics, chemistry, mathematics, and statistics, computer sciences, and of course, the only thing that remains biological is the biological system that one studies. Okay, when you study morphology, you start doing mathematical aspects to it. You want to do physiology, you start looking at the molecular interactions at each junction in the biological system, and so on. So you don't limit yourselves to just knowing the biological system by morphology and anatomy, or even physiology for that matter. You start characterizing it in all possible terms. Mathematics not excluded, physics not excluded, and also chemistry not excluded. So, why I am saying this at the outset is because it offers you entry even at an advanced stage. If you are pursuing biology, wonderful, you get introduced to biological systems at the beginning. But even if you have not started off as a biology graduate, you can try to connect yourself with biological streams while pursuing your baccalaureate or master's in chemistry or physics or wherever else, or biochemistry for that matter. Biochemistry is an early interdisciplinary thing that arose around 30s in biological pursuits. But today, if you have studied physics till late, you can still switch to characterizing a biological system and can be later, regarded as a biologist, biophysicist, and so on. The reason is that you know the realization of molecular phenomena underlying or underpinning all the biological processes that has unified the concepts of plant systems, animal systems, microbial systems, and so on. And this has also opened up new vistas of not only inquiry, but also of applying biological systems. You see, biological systems are self-amplifying, self-reproducing systems. So, it makes a lot of sense if you want to produce something through biological systems. They are programmed to amplify themselves. They are programmed to reproduce themselves. However, you should have the information in the biological system either introduced by you or naturally carried by the biological system that one is handling. So this is an advantage of biological systems. But even that is not a complete narration of today's biological pursuits. Biological pursuits today are not just limited to applying them to make industrial products and things of that sort. I have made a cursory, you know, for a cursory glimpse, I have made a PowerPoint presentation. I think I can share the screen with you on the PowerPoint. Vadidi, is that okay? I had initially thought that it will go with just extempore presentation.
Yeah, no problem, sir, no problem.
Yeah.
Please, Deepananda, can we give sir the rights to share?
Okay.
Sure, ma'am.
Yeah.
Yes, sir. Just give me a second. Yes,
Yeah.
so you can share now.
Also, sir, just a request, if you could adjust your audio also, please. It's a little unclear this side.
Oh, I see. I Okay, that causes concern. Is the slide visible? No.
No, not yet.
Use the want to. Oh, where are we lost? Oh, if I was better talking. Probably you did not allow me to share the screen.
No, we did. Deepananda, have we allowed, sir?
Yes, sir. You have the access.
I have the access.
You just need to click on share screen at the bottom of your screen.
Now I don't even get the host disabled participant screen sharing. So what do I do here? I say okay, but you disabled my video screen sharing. I say share a screen now. Okay, and then I go to share. Career options in biology. That's the theme that we are working on today,
Now
right?
see it. We can now see it.
Yeah. Okay. Wonderful. So I will just what I will do is I'll just uh, pass you through lists of things, kinds of activities, and probably also your aspirations with your pursuits in biology, career pursuits in biology. And that should give you a slightly better idea of the possibilities that biology today offers you. I think this should not have been the very first slide, but if it has come, I will just say there are options of pursuing biotechnology more often than biology today. And excuse me, there is a phone ringing. I should take it. Now I have muted my phone. It should not disturb us during presentations. Yeah, so you have possibilities of pursuing biotechnology right away at BSc level. There are hundreds of colleges that are offering this, but I am not pushing you in that direction at all. You should quietly and coolly pursue what you are doing. It's important to have solid foundations. Then, just jumping onto doing applications right in the beginning of your career. So whatever you like most, pursue that. because biology offers you linkages till late, as I said. Okay, uh, so, but if you are right away trying to start off with biotechnology, there are hundreds of colleges which are offering biotechnology programs at baccalaureate level. But again, these are education stops commonly. Don't rush to any or every of them that uh, are listed probably as biotechnology institutions. Okay, so I am just advising pursue what you have in a good institution with good syllabus and good teaching programs. And I'm afraid I would not very unhesitatingly ask you to give first option for private privately managed institutions. Even today, most of best teaching institutions are among the universities, particularly the central universities like Delhi University. Jawaharlal Nehru University doesn't offer BSc programs, but that's a good institution as well. You can have Hyderabad Central University among your preferred institutions. But again, not everybody can be accommodated in these institutions. Obviously, you have to opt from among institutions which are in private sector, privately managed institutions. So don't hesitate. But I'm just saying that you should be choosy of which institution, which program you want to opt for. Okay, and you don't have necessarily to take a jump uh, at the outset in the beginning itself of your career. You can link yourself with biology uh, at the postgraduate level or even at doctoral level. Postgraduate meaning master's level, and I'm sorry, graduate has different connotation in the West and East. Uh, then one thing that you should be clear about is where do you want to be in longer run of your career? You want to be in academia, doing basic research and teaching. That is one aspiration you can nurse. You want to pursue medicine. You want to apply new tools in diagnostics, prophylaxis, and therapy. This is what defines medicine. You want to be in industrial sector, large scale production of products of general biopharmaceutical or agricultural interest, or you want to run your own enterprise. You must evaluate yourself on this. No one can advise you. No one can instill in you the choices, because your liking are your likings and your decisions you should own, and you should try to follow those. Okay, but carefully. You should have some degree of flexibility. You should always have a fail-safe program if you are, for example, pursuing academic research till advanced stage, and you are offered by a pharmaceutical giant like Abbott or another Boehringer Mannheim or whatever you have, then you should consider it carefully as to what it fetches to you in terms of returns, and whether it also has projects that interest you. Having money and not having interest in the projects also is very frustrating experience in career. Okay, so I'm just asking you to be careful while choosing these streams that you want to be in. Pursuits in biology, this I had stated, they unify all disciplines in many ways. Many ways here. Uh, Biology today is interdisciplinary without any questions. It is multidisciplinary very often. It just does not, hobnob with other disciplines and come back. It very often requires intensive inputs from physical sciences. Okay, the conventional biology, of course, has had biology as an art in the beginning, and then biology became a science in 20th century, and it evolved to become a multidisciplinary pursuit. That is what modern biology is. The realization of the genetic material and the information transmittal in biological systems, that has let me probably magnify this a little bit. Uh, that has opened up possibilities of not only having interactions with mathematics, physics, chemistry, and whatever you have, but also to think of applying the molecular tools into biological systems and produce what you would like producing. Okay, and as I said, biological systems have the advantage of being self-propagating programmed systems. You can program them genetically and let them amplify themselves and produce your material or your chosen molecule in quantities that you would like. Okay, the challenge there is when you want to express a molecule, chosen molecule like interferon in a heterologous system. Interferons are produced in mammalian systems. They are antiviral proteins. You want to produce them in bacteria or in yeast or other systems, then you may have to tinker the genetic information to optimize their expression in heterologous systems, because heterologous systems have their own, nuances, like the codon preferences, their transcription translation nuances, and so on. So you will have to optimize that. But once that is done, you have a programmed system which is expressing your molecule. Basic biological phenomena unify at the molecular level. All the genetic information in cellular systems, which prokaryotes or eukaryotes, they all have DNA as the genetic material. That is the informational molecule, and the functional molecules are proteins and RNA, which are equally significant. RNA today is known to offer lots of epigenetic modulations of gene expression, and that is something which must also be of interest to you if you are advancing with biological sciences as your career option. Now, major orientations, I would even, before doing this major orientations, which you are all familiar with, I would say, what do you want to have from biological systems? If you are inclined to apply biology, information in biology, you want to have the same things which our ancestors, ancient ancestors want. You want to have good food, ample of food. You want to have good fiber, good fabric. You want to have fuel to run most of your systems, and so on. And these are, produced through exercises which are part of agriculture, animal husbandry. Environmental sciences, of course, has become a concern today because we have been, using fuel at an uncontrolled rate and have been emitting lots of partially burnt up fuel in the environment, and that has caused a lot of problems. We are doing chemical industries, and that is emitting other kinds of pollutants. We are using pesticides, herbicides, and this kind of uh, substances, which finally, end up in the environment, and many of them are recalcitrants. They stay for years in the environment. So, a concern for you would be to keep the environmental quality acceptable for good health. And that is where environmental sciences come in a big way as an option. And biological systems can be used to meet some of the environmental concerns that we have today. You may have directly an interest in industry or industrial product. You want to be an entrepreneur, want to produce something, and there should be a possible biological system for you. And there is plenty of information to optimize production of molecules of your choice to your chosen biological systems. And then, of course, health-related substances that you may like to produce. You may like to produce, vaccines, for example, prophylaxis molecules. You can do this. A lot of this today can be done in silico, and you make a short list before launching your experimental pursuits. Finally, you have to do your experiments and produce the molecules in biological systems. But initially, I mean, why initially? I think in our times as well, 1980s, when we were passing through research, to make a vaccine would take you 10 to 15 years. from conceptualizing to actually making the clinical application. Today, one can make promising vaccine molecules within matter of 6 months or a year. Of course, what still cannot be circumvented or bypassed is the need for phase one, phase two, phase three clinical trials before releasing the vaccine material for use, excepting in the situation that we have just passed through, COVID, for example. It has necessitated that you make emergency release, emergency use of the promising vaccine molecules. And the vaccine molecules, as you have seen, are not just proteins now. You can also have coding sequences for a protein. You can deliver it to the system, to the vaccine, and the vaccine produces inside body the coded protein. I mean, the spike protein of COVID-19, as you know, has been commonly used as a vaccine material, and its coding sequence in terms of, in the form of stabilized RNA molecules is what was used initially for vaccination. So this is just illustrative. By no means the list that I'm giving is comprehensive. Okay, I'm just illustrating what can be your orientation, what can be your career choice, what can be your aspiration, and what kinds of molecules you may like to do. Now, as you graduate, as you advance, take a few steps, you may find yourselves drifting towards molecular biology, molecular genetics, and genomics today, rather than just knowing the chromosomal constitution of a biological system. You may be interested in whole genome sequencing. You may be interested in genome-wide expression profiling of the system, and so on. That is what one calls genomics. Cell biology today, a lot of disease are associated with loss of cells, as in case of neurodegeneration, for example, or gain of cells, unwanted cells, as in case of neoplastic, diseases. And you can make use of cell biology for replacement of lost cells, certainly. Of course, when you have to, control the growth of cells, then that involves another approach of trying to induce a built-in program in the cells for causing their own death, cell death programming, or apoptotic approach for you. If all cancer cells can be induced to, undergo cell death, you will be able to treat cancer. That's one common approach in cancer. But of course, you see that as the cancer advances, you cannot wait for the system to adapt to induce or reprogram its death, but you resort to chemotherapy, radiotherapy, radiation therapy, and so on. So that is a different story. Depending upon the phase one is handling of a disease, one can have one's strategy for therapy. You may have immunology, for example, modulators of immune cells, and you may use the dendrocytes that are, involved in antigen presentation, antibody production, and so on, or natural killer cell biology for that matter. And you can enable the systems, the patient, to handle a challenge coming from an infectious disease on its own. Developmental biology, is, what it deals with. It deals with development of all kinds of cells from common progenitor cells. And this gets relevance as a basic issue, yes, but in terms of disease, you get lots of developmental disorders. And this can be your specialization. You may specialize yourself in cancer biology. Immunology, I have mentioned twice. I'm sorry, we have already talked about it. Endocrinology, the production of hormones and their functioning and dysfunction in several diseases. Neurobiology, neurophysiology, that is an upcoming discipline also. And this can be your interest. You may be interested in parasitology, protozoan and other kinds of pathogens causing disease in humans. Microbial pathogens, bacterial diseases, viral diseases, and so on can be of your interest. Virology can be your interest, just studying biology of viruses. Plant cell culture and regeneration of whole plants from cultured cells or calli. This can be your interest. Molecular signaling. You know, biological systems are equipped to sense fluctuations in the environment and also within themselves. I mean, inside the biological system itself, there can be environmental cues to signals, and there can be an innate cue to signals, and then the system reprograms itself to deal with fluctuations in environmental stimuli, environmental factors, or to respond to any signals coming from inside the system to take a certain course of development or a certain course of release of certain molecules in response to that. The entire developmental biology, as a matter of fact, connects with signaling. So also immunology. I mean, signaling is not a separate islandish discipline. It connects itself with more or less everything that we have talked about so far. You may be interested in bioinformatics if you're, this is what one calls dry uh, activity, dry research activity. You are on a computer, you have lots of data of biological systems, you have lots of information coming on, for example, of an epidemic, the virus's genome sequence, and so on. And you start doing what it codes for and what the molecules will look like entirely in silico. So you are actually not doing experiments, but I mean, what you do on computers is also is a kind of experiment. And so you arrive at what are the most potent molecules, which will be most promising molecules, which will be therapeutic targets, and what will be the most promising vaccine material, and so on. So that's where you do bioinformatics. Your interest may be in RNA therapeutics. In recent times, RNA has emerged as a major regulator of gene expression. And so RNA interference is a phenomenon which might interest you. You may be interested in ribozymes, for example, catalytic RNA, antisense RNA, siRNA, small interfering RNA, or microRNAs, or long non-coding RNAs. These are some of the kinds of RNA molecules that variously influence gene expression. Your interest may be molecular evolution and origins of life. Very basic question, fundamental question. How did biological systems evolve, originate, first of all, and then how did they evolve? This can be your interest. So, and even this list is not comprehensive. It is just an illustrative list of the kinds of activities towards which you might specialize yourself depending upon your interest. Uh, so far, is that clear? Is that making sense in the audience? If there is a problem, please raise your hands, and I think the questions will be conveyed to me.
Yeah, so students, if you have any questions or you want to speak to Dr. Pramod, please use the reaction button and raise your hands, and we will unmute you and allow you to speak. So you can please carry on, and we will convey it along the way.
There are some hands raised there, Kushi and Krishnamurti. So the other way is that I have just 4 or 5 slides to give you. And so after that, I can take all the questions, or I can as well take Kushi, Krishnamurti, and Om Asutosh. Since we have limited time, I think I finished what I have to say.
Yeah, and so you please, yes.
Keep your questions, reserved. Now, the skills, common skills that one acquires, the techniques and methods that have come up. Now, I must just state that biological research today, or biological studies in general, have become very technology intensive. And that also makes them capital intensive in a way. So you have big budget projects usually, which will be frightening to an uninitiated person, but they are manageable. They produce materials far in excess of the value that goes in their research phase or development phase. The skills that one requires or acquires while pursuing biological sciences today include cloning and expression of proteins of your choice, genome-wide gene expression profiling, making of siRNA, miRNA. siRNA is small interfering RNA or microRNA, and long non-coding RNAs. Their biology and applications you study, and then you visualize associations of genes that express. If you biological systems are characterized by a homeostasis. So if you have a number of genes that are expressed in a system, if you enhance expression of one gene, it is likely to influence another gene's expression in a positive or a negative manner. So these are called associations of gene expression. So you also want to profile associated gene expression. So you visualize the association. Biomolecular structure modeling, you want to see what is the structure of a molecule and what will be the cognate structure of another molecule. See, supposing you have defined a therapeutic target for yourself, then you try to interact. This is all in silico. You design, you model this structure and try to interact it with small molecules. And if small molecules are docking with great stability with the therapeutic target, chances are that they will turn out to be therapeutic molecules, the smaller molecules. Okay, and that becomes a common concern in medicine. But that is not all. You want to see, what a certain sequence of DNA will interact with, for example, a certain protein. So which phase of the protein is interacting with which sequence of DNA, and what is the consequent influence on the biological system or on gene expression in this system. Okay, this can be a concern. So you acquire skills for biomolecular structure modeling, and then you have to testify it by experimental verification. And the biomolecular modeling is done by molecular dynamics. There are programs available. You have the primary sequences of amino acids in a protein given, and then you model it to make a three-dimensional structure. And then you experimentally determine whether this structure is experimentally tenable or not, testified or not. So you do X-ray diffraction studies or NMR studies, nuclear magnetic resonance studies, and that gives you the experimentally determined structure. So you do a lot of bioinformatics, as I had said earlier. Bioinformatics, because the data is burgeoning. There's now with genomics coming in, the rate of generation of data has enhanced by leaps and bounds. If it is told to you that there are terabytes of data, most of the common computers would not take it for processing, but terabyte of data is very common passage for you in today's biology, in terms of genome sequence, in terms of gene expression, and so on. So you have to have skills in bioinformatics. As a matter of fact, bioinformatics also connects itself with most of other pursuits. So while you are doing cloning expression and other things, you also are visualizing bioinformatically as to what sense it makes through the database in the public domain. You may have interest in drug discovery and drug design. Drug discovery because you find common patterns of a certain molecule occurring in a certain physiological state, for example, a disease state and a healthy state. In that case, you will like to, pinpoint on possible role of this molecule in disease or in a certain physiological state. Okay, physiological states can be varied, not just disease. It can be that you are passing through stress, you are starving, you are having, starving by option. God forbid, no one should starve. But the fact remains that a certain section in the society does not get enough food for feeding. This is reality, even if we produce enough. But let us not go into that direction at all. What I'm saying is physiological and other kinds of variations, physiological states can be, you are subjected to heat, you are subjected to cold, you are subjected to hypoxia, you are subjected to hyperoxic conditions, you are exposed to a certain pollutant. So all these situations can be profiled for expressions of certain molecules. And if you come across certain patterns, you start thinking of it as having a role in that particular physiological state of the organism. And so there you start doing possible drugs, possible drug targets, and so on. And yeah, I think I have a tendency of talking too much in drug discovery. Uh, why is it important? It's important to realize. You see, initially, when we did not know enough about systems, we had hit and miss as the only option. You have a disease, you give it all kinds of things, and some of those things might work in that disease situation, and you pick that as a drug. Uh, this can't be a continuing experiment. When today you have billions of people on Earth and the millions requiring a certain kind of assistance, therapeutic intervention, you can't just start trying different variants of molecules and think that you will finally be in command of the situation. You can't. You have to have expeditious approaches to finding out drugs, drug targets, and then you know you proceed accordingly. Even that at times is very daunting. You don't get very timely release of the drug for various reasons. So how do you rationally design drugs? You visualize its structure, the possible therapeutic targets structure, and then you start screening for cognate molecules of a smaller molecular weight, and make a short list of possible drugs, and then test them experimentally before launching in the clinic. Genomics today, I think this can be, your interest can be just characterizing genome of an organism. That is the most ordinary or primitive application of genomics, that you want to know the sequence of the entire genome. Uh, but then you may be concerned with functional genomics, that means expressed subsets of genes. And your interest may be in archaeology, in paleontology, and in the archaeological materials, you may try to amplify DNA and start correlating it with possible course of migration, possible course of interrelation among populations that lived in Egypt and that lived in Mesopotamia, etc., or that came to India finally and settled down. So one can trace the course of migration, etc., through genomics. One can trace similarly the nature of flora and fauna, maybe millions of years back, if you get suitable fossil material from those periods. Okay, so you can have paleogenomics as your interest, you can have the archaeogenomics as your interest. You can also have sociogenomics. I have not listed it here. You may have various socioeconomic strata profiled for expression, and you might correlate it with what is actually causing a limitation in a certain section of the community. It is not being fed properly, it is not getting a particular supplement that it requires, or there can be several other reasons. Okay, so sociogenomics and ecogenomics, ecological variations in terms of habitat, they can also correlate with gene expressions and profiles. The offshoot of the genomic approaches has been next generation sequencing protocols that enable you to do, whole genomes, whole human genome, for example, in a matter of a day or so. People are trying to further accelerate it, but this is possible now to have entire genome sequenced within a matter of a day, and so you can take quick clinical decisions. Earlier, it was not possible. The first human genome took 13 years and $13 billion to sequence. Okay, and that too was a draft sequence. Today, it is possible to sequence the entire human genome within a matter of days, and at an affordable price. They want to bring it down to $1 per genome. It is not yet there, but it has come closer to $100 per genome, per human genome sequence. Proteomics and metabolomics are other kinds of skills that you may acquire. Proteomics involves profiling the entire protein constitution of the organism, which proteins are involved. Essentially, it involves mass analysis, mass spectrometry, after making fragments, peptide fragments of the proteins, and making them charged, charged by ionization or otherwise, and then releasing them and seeing what fragment patterns are produced. And that gives you a fair idea of what kinds of proteins are expressed in a given situation. Then you may have instrument mediated instruments like confocal microscopes. You want to do live cell imaging, you want to see the cells live. You may like to do in situ immunocytochemistry by fluorescence microscopy. You may be interested in sorting cells. We have several hundred types of cells in our body. The blood itself has several dozen cell types. You may like to sort them. You may like to enrich fractions for a certain cell type. And that is achievable by flow cytometry. Cryo electron microscopy goes to the resolution of 2 to 2.5 angstroms, and that enables you to see the molecules in their three-dimensional structure while they are engaged in a certain function. For example, molecules that make a pore, nuclear pore, and the molecules that interact with the pore proteins, they can be visualized in real time, in action, as a matter of fact. But now, for visualizing, you have to freeze them in a certain state, and then you visualize. So 2.5 angstrom resolution, something which was considered to be a great X-ray crystallography some time back, is attainable by direct visualization. You are not doing crystallography, you are actually visualizing the molecules in action. Okay, that is what cryo-EM involving tunneling electron microscopy has enabled you to do. Viral reverse genetics, this is an approach that helps you produce viruses with their genomes defined by you, not by the naturally occurring viral genome sequences. This is something which has taken a wrong direction. The conspiracy theorists, they blame China for having indulged in some kind of a viral reverse genetics to produce, infectious virions. But I must tell you that this is a very promising area which enables you to produce targeted delivery vehicles, targeted delivery vehicles for drugs, targeted delivery vehicles for vaccines, and so on. So viral reverse genetics is not something to shy away from, but of course one should be careful. It can take a direction which can be hazardous, like if COVID really was produced as a bioterror tool, as a weapon to biologically destroy populations. I think that is a conspiracy theory. Don't worry about those right now. I am not a particular contributor to that theory at all, but I don't rule out the possibility nevertheless. Okay, you can today do gene editing. You know, Jennifer Doudna and Luke Charpentier, they developed a system from the CRISPR-Cas system offering in microorganisms, and this can be introduced into higher organisms, and they can make targeted correction or modification of genes. Okay, and this has a great promise and potential of dealing with genetic diseases. Prenatal and postnatal treatment of genetic disorders can be done employing the CRISPR-Cas tools. There are other tools as well which help you edit genes and genetic sequences. There are stem cells for cell therapy. You can have lost cells can be your concern. So if you have stem cells somehow rescued and amplified in culture, I get that there is a power off here. Do you get my voice still?
We do, sir, we do.
You do get, okay. But I got a signal that there is power off here. So I'm putting, uh, am I audible now?
Yes, yes, sir.
Okay, so I continue talking. So you can have stem cells for cell therapy where loss of cells is a concern. Uh, you can also be interested in producing transgenic plants and animals for producing something of your interest, for enhancing yield of what the plant is already producing, or for introducing an extra flavor to the product, for producing long staple cotton, for example, cotton producing longer staples than its usual length. That will be a lot stronger fiber and will be a lot stronger fabric. Uh, what has also come in, made a lateral entry is artificial intelligence and machine learning in biological systems. And in biomolecules today, commonly they do machine learning because it is not possible to manually handle all the data. So they invite machines to do certain kinds of analysis based on the data available in various databases. Let me see what else I have. Emerging applications in biotechnology. Again, we are talking of biology. Keep in mind that biotechnology is just an offshoot of biology. You can have unraveling of history, paleogenomics, as we talked of. The fossils characterized for sequences that they have. Ecogenomics, environmental variations as determinants of gene organization and function. Sociogenomics, social stratification and gene expression. Ex vivo gene therapy, that can be your interest. Gene editing can be your interest. Three-dimensional printing to produce organoids and organs can be your interest. Bioethics is another thing. Since there are so many promising, new tools, there are also equivalent or proportionate number of possible hazardous directions that they can take. Okay, that is why it is important that all these approaches are very closely monitored and regulated by institutions. So bioethics is one thing. Certain kinds of activities are totally banned. Certain kinds of activities are allowed. And this has a certain principle of which activity will be allowed, which will not be allowed. For example, gene editing is one thing which even though it is very promising, it is not allowed for directly being applied in human embryos, okay? Because this will be playing gods. It's a concern similar to what happened in recombinant DNA technology when it was initially discovered in the early 80s. They made very stringent control. Similarly, gene editing right now is under very stringent control of regulations. However, ex vivo applications of gene editing are allowed. So you can have biopsies from patients, culture them ex vivo, modify them by gene editing ex vivo, and reimplant them in the system so that the lost functions are kind of made up for. Okay, so bioethics is a very important thing, and this is not just for gene therapy, it is also for other activities involving biological systems. Then a concern, as some of you may be interested in law related to all these regulations and rules. Intellectual property right is one important area where a lot of our students, earlier students, have digressed, and they are just practicing IPR related issues. And this can be of interest of some of you, at least, I guess. Okay, so this was an illustration of all kinds of possibilities that one has with biology. Now, I would open up to take your questions. There are, quite a few questions that Varidhi had given me about what are the possibilities and whether one can have we can be back to our full screen.
Yeah, the Pandita, can you please stop sharing? Yeah, thanks.
Okay, so the institutions in terms of, there are many.
that's not limiting at all. But prospects of pursuing a supported higher study in biology, that is something which remains to be addressed. Most of the PhD pursuits abroad and also in India, I must say, they are duly supported by fellowships. You may have to pass a certain examination, like here you have NET and such examinations which qualify you, entitle you to draw research fellowships. And that would enable you to pursue research in area. But for BSc and MSc, there are not many fellowships in India or even abroad. The things become somewhat doable in the West because they offer you assistantship of all kinds. So in the departmental activities, you may participate in a small teaching, or you become a laboratory assistant part-time, and you are pursuing your study. So that kind of compensation sometimes comes handy. But in our country, we are lords of our own wishes commonly. We don't have many options of working while we are learning. So this is a certain impediment at the level of BSc and MSc, but at the level of PhD, even here, we have lots of fellowships offered by DST, DBT, CSIR, UGC, ICMR, ICAR. These agencies, they offer fellowships to pursue research. So you can definitely think of, winning a fellowship for yourself and pursue your higher studies. Let me now get back to the audience and take questions, because that will otherwise remain.
Yes.
It's 5:54. Yeah, we can continue. Varidhi, how sharp is the time limit?
We have some time, so we will take a few questions. Students, if you have any questions to ask, please raise your hands using the reaction button, and we will give you a chance to unmute. So we have a couple of hands up. Yeah, Eshna, can we please unmute Eshna Debanata?
Yeah, ask your question.
Good evening, sir.
Yeah, Eshna, yes, please go ahead.
Sir, actually, I wanted to ask about BSc Neuroscience.
I don't get you clearly.
BSc in, sorry?
She meant BSc in neuroscience.
Okay.
BSc in neuroscience.
Yeah.
I don't know of any institution offering that kind of a specialization, but MSc with neuroscience specialization certainly is offered by a few institutions, including JNU, but JNU does not brand it on their degree. It just gives you degree in life sciences, MSc degree in life sciences, but you have neuroscience papers that you can take as your advanced course. Okay. Similarly, Gwalior, I think, has something in behavioral sciences. It's a state university, and it is, a lot of it depends on the person who is coordinating the program. Right now, the person coordinating in the program is a behavioral scientist, so he gives this option. But I wonder if it will continue forever. So neuroscience at BSc level is not very, very common. As a matter of fact, no institution classes to my memory in the country which offers you BSc in neuroscience. BSc zoology, but if there is a neuroscience specialist in the department, it gives you a reasonable exposure to neuroscience. Okay. And master's zoology, many departments have neurophysiologists, neuroscientists, so they give you a reasonable dose of neurosciences. Okay.
Yeah. Okay. Thank you, sir. So the next question will be from Om Ashutosh.
Om Ashutosh.
Yeah. We're just unmuting him. Anji.
Hello, sir.
Yes.
Am I audible?
Hello.
Yeah. Yes, Om, you're audible. Yes.
Sir, I wanted to ask a question on environmental science.
Yeah.
Can you light up more about it?
Can I illustrate what are the possibilities?
Yes, sir. That is what he means.
Yeah, when we are polluting the environment, we also want to depollute it, right? So anti-pollution drives, they demand development of certain kind of technology, like our effluent waters from residential complexes. They can be two kinds: those with lots of organics, those with not so much organics, like the bathroom effluent and the kitchen effluents don't have so much of uh, BOD, but the toilet effluents have high BOD. So modern, residential projects must have their own built-in sewage treatment plants. Okay, and sewage treatment with rapid recovery of water quality. That's the challenge. It's not there in most of the modern residential projects, but that should be the goal. We should have all our effluents treated to bring back them to good quality, and of course ideally water treated back to become potable grade. That is the dream. I can just mention to you, I was in Zurich 1980s, 86, 88, 89. There the population is much smaller, so I must not just say that everything that they do is possible to do here. Delhi has a population of 1.5 15 million people or 16 million people, and Zurich had a population of 300,000, not even a million. So they would treat all their water, all their effluents coming from all sources in one place, and would recycle them at potable grade. Potable grade means not having any heavy metals, beyond the permissible limits, of course, tolerable limits, and having no BOD that alarms. But they would not pump it back into your water lines. They will just discharge it into their river called Limat. And so Limat is carrying pure water. What we do, we just discharge all our effluents in the river Jamuna or Ganga, wherever we are, okay, or Kaveri for that matter. And so this does not, give a sense of very civilized and conscientious living. So we want to have, anti-pollution technology. We want to have bioremediation, for example, remedying, materials or soils, for example, dosed heavily with nickel, cadmium, mercury, and so on. If we can sequester those metals from the polluted soils, that will be a day to celebrate. Okay, and there are technologies which are now being available, employing biological systems, particularly microorganisms, which help you remedy some of these heavy dosing of pollutants in certain parts of the environment. So that is what I meant by environmental, and then environmental technology itself. For example, para-environmental technology, if you develop a technology which helps you produce biodegradable plastics. Poly-beta hydroxybutyrate is one such material that is biodegradable and has all properties of plastics otherwise. So that can be adapted to be produced at a large scale. Otherwise, if you can develop organisms which can take polyethylene, polypropylene, and polyamides as substrates for their growth, that will be wonderful. So far, we don't have such organisms in commonplace. Okay, but there are possibilities that you can develop plastic-eating microorganisms. That will be an environmental biotechnology for you. Then there can be chemical technology that helps you also recycle the polymers, polyethylene, polypropylene, polyamide, or polyesters of all kinds. And that will be helping you keep environmental quality in control. Okay, so these are some of the directions that one can take if one is environmentally conscious. Did I answer your question? Partly, yes.
You have, sir, but I can't find the home again. So we'll take the next question anyway. Sai Teja.
Good evening, sir.
Good evening, Sai. Yeah, ask your question.
Yeah. Sai, please ask your question.
Sir, I wanted to know about pharmaceutical engineering.
About
Pharmaceutical engineering.
pharmaceutical engineering. Hey,
Yes.
pharmaceutical engineering. You see, what do? You choose drug targets, you develop drugs, and we were talking of drug discovery, drug development, drug design. Okay, these are activities that one can undertake. Now, a lot of it comes from basic research in biology. For example, what are disease-associated genes that are expressed in a given situation? These can relate to infectious disease and non-infectious disease as well. Can be cancer disorder, can be neurodegenerative disease. You can have other metabolic disorders like diabetes mellitus or developmental disorder like rheumatoid arthritis and so on. Geriatric diseases you can have. Now, each of these diseases is characterized by overexpression of certain kinds of molecules or expression of a set of molecules in certain proportions. Now, you choose them as possible therapeutic targets. Either you enhance their expression to bring it to physiological normal concentration, or you have to impede their accumulation in the system. I can illustrate this. We had a ribozyme which will target TNF alpha, tumor necrosis factor alpha. Tumor necrosis factor alpha in rheumatoid arthritis overexpressed in the joints, in the synoviocytes. Synovium means the cushion material in the joints that you have. Okay, so TNF alpha, if you could target it and bring its concentration down, it can help in mitigating the symptoms of the disease. And we used a collagen-induced arthritis model, a mouse model, of course, not human, and demonstrated that if we express our ribozymes in the mouse, then the onset of collagen-induced arthritis is delayed. Okay, this is just illustrating one point, and that too is a kind of disorder that is innately produced. Okay, not an infectious disease, not cancer, and not a metabolic disorder. So each disorder is characterized by expression of a set of molecules in certain proportions. You see, and this is what the healthy individuals show a certain profile of expression, and the unhealthy individuals show a deviation from that. In fact, easy state and disease state are characterized by differential expression patterns of different molecules. Okay, and if you have information, plenty of information on that, you can choose some of them as potential targets for your therapeutic strategy. However, that is not yet pharmaceutical engineering. Pharmaceutical engineering starts with optimizing the mass transfer and energy transfer aspect of a process. You are producing a medicine or another industrial product. Of course, you said pharmaceutical engineering, so pharmaceutical engineering produces medicines, pharmaceutical materials. So there you are optimizing the mass transfer and charge transfer, energy transfer efficiencies, thereby making the process economical. Okay, and that is what optimization of biopharmaceutical processes is, or any industrial processes. It optimizes mass transfer and energy transfer aspects. You can, engineer new strains of bacteria or other microorganisms that are using for that particular product. Just an illustration, not biopharmaceutical though. In brewing, you have, most of the yeasts, they will tolerate ethanol concentrations up to 6% or so. The best of the strains, not the milder ones. Milder ones will start dying at 3%. Okay, but you select for those strains which can tolerate 10% ethanol. Now, what difference it will make? It will make a difference that you can, in one brewing step, you can go up to 10% ethanol concentration, which will be a very good beer, for example. Beer is unfiltered, undistilled brew. wine is undistilled brew. So if, of course, it has post brewing, it has certain processing of filtration and maturing and all that, seasoning. So you can have strains which are suited to take you to that kind of concentration in one go, or you can start using a new substrate for making beverages with a certain vitamin content to it, or with a certain pharmaceutical content to it. And that is part of your biopharmaceutical engineering. Of course, this also covers the visualization of molecular structures, visualization of structure of cognate molecules, and so on. And you advance yourself in the direction that you want to produce a certain material in a certain quantity by a certain process. Okay.
Okay, so sir, we'll carry this on for another 10 minutes and try to take as many questions as we can. The next.
You can carry on. I am free this evening. I have long this time. I do not know if you are limited on your time.
We'll take some more questions, sir. Next would be Megha.
Megha Gupta, yeah.
Good evening, sir.
Good evening, Megha.
Sir, I want to ask about BSc Anthropology.
About what?
BSc in Anthropology.
BSc in Anthropology. That's a very good question. I had not illustrated anthropology. Anthropology is a science dealing with humans, right? It includes human development, biology, behavior, and many other things. BSc Anthropology, I think the Department of Anthropology in Delhi University does offer this. And there are other institutions in South. I think there is one in Madurai. Kamraj also has a component in behavioral sciences and in anthropology. But I will have to recheck if they have BSc in anthropology or not. Okay. Do you had a component of anthropology in BSc earlier? Okay. And of course, anthropology is something which has drawn attention of late, much more than in past. I'm sure new departments will come up offering anthropology. Established department, I don't know many that offer anthropology program formally at BSc level. At postgraduate master's level, I'm sure there are many other departments. There is one in Hyderabad, there's one in Delhi, one in Madurai, and let me see. Pune, no, Pune, that's not anthropology. That's not anthropology. In Mumbai, there was one institution which dealt with Tata Institute of Social Sciences, probably was introducing anthropology paper, not a program in anthropology. Yeah. So that's your interest. You know, anthropology, a component of it, important component of it is psychology. Commonly talked about component of anthropology is psychology. So you can orient yourself, keep connected with basic biology, genomics, and psychology in a longer run, not at the BSc level, but in longer run, if you end up doing research, you can think of linking yourself with genomics and probably find interesting gene sets that are expressed in different behavioral classes of humans. And of course, archaeology, as I said, archaeology connects very well with the genomics. So also paleontology connects with genomics. Okay. Your question was institutions. So I must get back rather than illustrating the academic component of it. I should tell you that there are a few institutions definitely offering anthropology at master's level. BSc anthropology, I do not know many institutions.
Okay, so the next question will be from Aryan.
Aryan, yes.
Hello.
Hello, Aryan.
Yes, sir, I am audible.
Yeah.
Sir, I pursued my BSc Biotechnology course. But sir, I want to know that what are the skills and the things we should undergo when we are studying the biotechnology? Tips and some tips when we are studying it.
Can you probably be a bit louder? I'm maximizing my volume here at this end. Yeah.
Hello.
Repeat your question. Ask.
Sir, I am telling you that I am pursued my BSc biotechnology course, and what are the tips and tricks we should undergo when we are studying it? What are the skills or things we should focus on?
What should you focus on? This should be your decision. I just gave you a list of various kinds of things that you could do. You know, your interest can be in, for example, in neurosciences or but neuroscience related biotechnology, you will have to imagine for yourself. Okay. Biotechnology is not just bioprocess engineering. Okay. Bioprocess engineering is something that is closer to chemical technology, again, concerned with charge, energy transfer, mass transfer, that kind of aspects. But to design molecules, to make genetic engineering, and to make an expression system where you can produce these molecules really in good amount, that is what your concern should be. So if your interest is, for example, cancer related drugs, you can screen for what are the drugs that reach out to cancerous tissue, and will probably start eliminating them from the system. I wonder if I'm taking the direction that you wanted. If I am not clear about your question, you can stop me. Okay? Aryan? So I was just trying to illustrate that your interest can be, I mean, you define your object, what you want to do. And that you do eventually at the research stage, not while you are pursuing bachelor's or master's programs. But in master's program, you do have to orient yourself for a certain direction. Whether you want to do genomics of general nature, you want to do a functional genomics for a certain purpose, whether you want to find drug targets, or you have already defined drug targets in mind, you want to find the drugs that will interact with them. This is a very popular activity in biopharmaceutical field. BSc biotechnology should also get oriented in this direction. All you need to have is bioinformatics skills, and you choose some of your drug targets and start offering it the possible drugs that will interact with this. The principle of medicine is that whatever is overexpressed and is causal to disease, that becomes your preferred drug target molecule. Okay, once you have defined that, you want to know what will inhibit it such that it will not let the disease advance. So you want cognate molecules, a molecule that will recognize this. Okay, not just recognize it, will disable it from causing disease related processes. So you choose your drug target, design your drugs, and dock them in silico. Okay, do molecular dynamics and see the docking energy and other things. And then make a short list of molecules that will probably inhibit this drug target from doing what it does in disease situation. And then from that short list, you can further shorten and finally synthesize a few of those molecules. You see, drugs should preferably be small molecules because they can be delivered in high concentration but small masses. Their small mass will become a larger number of molecules, right? But if you have a large molecule, then and you want to give a large number of such molecules to the system, then there is there is an internal conflict of the two criteria, because we have about 50 to 60 trillion cells. The adult human body has about 60 trillion cells, and that the irrational therapy that we use, we send out 10 to the power of 15 molecules per subject. That means 1 million molecules potentially on an average per cell. And that is not very logical system of medicine. But since we don't have alternatives developed yet, we are taking that. Even a painkiller or an antibiotic or other things, we inundate the system with the large number of molecules. Okay, and the rational drug design phase has defined drug targets, defined delivery of the molecules to those cells which need a treatment. Not all cells undergo a change in each disease. You have a subset of cells only. I mean, in a disease, if only 1 billion cells are altered or having altered patterns of expression, why do you want to target all the cells? You need to reach out to those billion cells. Now, what are these billion cells doing? They are expressing a molecule X. Now, you want to design a molecule that will recognize cells which are expressing molecule X. So you make a targeted delivery vehicle, and these vehicles can be non-biological nanomaterials or biological nanomaterials. You can have viruses that recognize such molecules expressed on the target cell surfaces. Okay, and then you have already narrowed the number of cells that you want to reach out. Now package these vehicles with the drug that you want to deliver, and the drug you have already chosen based on the recognition of the target molecules inside the cells. And that will give you 1,000,000 times reduction in dosage. If you are taking 10 milligram per kilogram body weight of a drug today, and you have already defined now the drug target and developed a targeted delivery vehicle, you may reduce it to 10 microgram per kilogram body weight for the same extent of effect on the target. So that is one thing which is doable. Which is desirable, and a biopharmaceutical engineering program may also have a component on development of drugs of this kind. Of course, biopharmaceutical industry, once you are into the industry, mainly involves optimization of processes rather than development of molecules so much. Development is part of it, but you have process engineering more often than the development of a drug. Okay.
All right, sir. Thank you so much. Because of a time crunch, we will have to wind it up for today. But we do still have a few students who have kept their hands up. Is there some email ID that we can share with the students and they can reach out to you in case they have any questions?
Yeah, sure. I will respond to the questions when they ask.
Yeah. So could you just speak out your email ID? We'll put it in the chat.
What do you want me to do now?
If it's okay with you, can we share an email ID of yours with the students who want?
You can, of course, you can.
Can you just speak out your email ID? We'll put it in the chat for the students.
Yeah, I can speak out. It is PK Yadava, PKYADAVA, all small letters, 1953 @gmail.com.
Dipanita, can we please put it in the chat? Yeah. So we've done that. So any students who want to reach out to Dr. Pramod can write to him on this email ID. If you want to reach out to Careers 360, you can write to us on info@careers360.com. With this, I would like to wrap this up. Thank you so much, Dr. Pramod, for sparing your time and enlightening us with.
You're welcome. It is such a vast field. I was quite aware that we are going to overshoot the given time. As it is, I have just enumerated a few things. I have not gone into the subject matter at all. But I hope that there will be some hint for the participants on which way to approach about their concerns. They must define their targets, their objective, where do they want to be, what is their preferred area of activity later when they have passed all the studies, etc. I think many of them should be initiated in a direction that they were missing.
Absolutely, sir. Absolutely. And with this, again, we would, on behalf of Careers 360, heartful heartfelt gratitude to you, and we would like to have you again very soon for one of our sessions.
Thank you. Thank you.
Thank you. Thank you, everybody. Thank you, all the students, for joining us, and we'll see you soon. All the best. All the very best. Thank you.
Okay. Okay, Varidhi.
Thank you, sir. Bye.
I get out. Yeah. Thank you. Thank you all.